Control of heart rate PowerPoint. It's spec 7.8i and II. It follows on from the work we've done on respiration and on muscle structure. And we're going to look at how we can increase our heart rate and decrease our heart rate depending on the level of activity. Learning aims for this section are sort of how the heartbeat is controlled. We'll look at the role of the autonomic nervous system. We'll look at chemoreceptors and baroreceptors and what they do. And then be able to describe and explain the effect of exercise on heart rate. That's going to be an activity that you will do on a handout. Okay, so you've covered the heart before. You did it in the AS. AO1 school just to label a heart up. You should know how blood flows through the heart, so the sort of passage of it through the heart when it comes to the vena cava and leads out of the aorta. That shouldn't cause you any problems. And you should be able to talk about the cardiac cycle and the different stages, and then link that into the opening and closing of the valves and the pressure changes in the heart. If you can't do any of those things, I suggest you go back to Moodle and have a look at them, basically. Go and look at the work from the first year. It's all up there. Okay, so here's a heart for you to label, so pause now. And remember, there is a handout that you can download from the VLE that accompanies this video. So pause the video and have a go at labelling it. Okay, so we're going to start with the vena cava, and we're going to go down to the right atrium, then we've got our right AV valve, then we've got our right ventricle. Around the bottom of the apex of the heart is labelled, this is the apex of the heart here. And then we've got a ventricular septum which runs up the middle there, separating the two chambers. That's right. And then we've got a left ventricle. We're going to go up to the left AV valve, and then here we've got a semilunar valve here and semilunar valve here. Okay, so these are separating the ventricles and their arteries. Pulmonary vein here coming in from the lungs, carrying the oxygenated blood back to the heart. Left atrium at the top there. Pulmonary artery, that's the vessel, remember, that takes blood away from the heart to the lungs to be oxygenated. Aorta at the top, that's going to bend round into the aortic arch, which we can talk about in a bit. And again, we've got a semilunar valve there. Now, cardiac cycle, this should be familiar to you. It's first year stuff. There is a handout, there's a diagram on the handout that you can now annotate. I want to know what's going on at each stage and what are those valves doing. So pause now and do that. I start in diastole, just because I do. And diastole, heart's relaxed. We can see that there, the valves, the AV valves are open here, and the heart's slowly filling with blood. So that's when it's relaxed. And then we go atrial systole, and you can see the atria here are contracting, and the forces push those AV valves open fully, so they're nice and open. Semilunars are still closed, so these top are contracting and forcing the blood down into the ventricles. And then ventricular systole, we can see the ventricles have contracted and they are squeezing blood up and out through the two arteries, and we can see that the AV valves here and here are closed because of the increased pressure in these chambers. Okay, so that's a cardiac cycle, and that's kind of what you learn at the end of the AS. Now, building on that, we need to know how the cardiac cycle is controlled. Okay, and cardiac muscle is known as being myogenic. That means it's capable of generating electrical activity, and it does that from a section of the heart called the sinoatrial node, or you may know it as the pacemaker. So electrical activity originates in the SA node, or the pacemaker, spreads across the atria, travels to the bottom of the heart, to the apex of the heart, and then spreads back up the ventricles. All this links into the cardiac cycle. So as the atria contract and then the ventricles contract, and you can link it in. It can be controlled, or increased or decreased, in two main ways: by your nervous system and sort of hormonal control. So think things like adrenaline. Okay, this is a diagram you have on that handout, so you can just label it up first. Things will label the bits and pieces first, then talk about what they will do. So we've got a sinoatrial node, or we sometimes see it called the SA node, and that it sits here in the right atrium, a little area of tissue where the cardiac impulses or the electrical impulses are generated. And you know, theoretically, you could remove the heart, and you would little bits of tissue would start to generate their own impulse all on their own. And then these connect to the atrioventricular, or AV node. It's also in the right atrium. It just sits sort of above the ventricles there. And what you've got running, separating the atria and the ventricles, the atria at the top here, ventricles at the bottom here, you've got a band of non-conductive tissue. So you've got a band of tissue that runs across the heart that won't let the impulses go down. Then we've got things called Purkinje fibres. Used to be called Purkinje, so if you see that in other places, it's the same thing. And these run all the way to the bottom of the heart, or the apex of the heart, and they run back up the ventricles again, and they will transmit that impulse along. And here's the bottom of the heart, or the apex of the heart as we call it. And then we've got a little area called the bundle of His, or His, whatever you pronounce it. And it's a little section through that layer of non-conductive tissue that will allow the impulses through. It's the only section where they can pass. So, okay, so that's everything labelled up. Okay, now you should have these 3 little diagrams on your handout. And what we're going to do is use all of them to kind of explain how it all works, how heart rate is controlled, how those electrical impulses originate in the SA node, travel across the atria, travel down to the apex and back up the ventricles again. So that's what we're going to look at, one in turn, basically. Okay, so diagram A. So what we've got here is our SA node in that right atrium, and that SA node is a pacemaker of the heart. It's where those cardiac impulses are generated. And it basically releases waves of electrical impulses across the atria. So they come out from the SA node and they travel across the atria. That's the first diagram. Okay, secondly, what you've got after they've left the SA node and travelled across the atria, you have got this layer of non-conductive tissue sitting here, and just by is the AV node. Okay. Now, the impulses all, once they've travelled across the atria from the SA node, they all gather at that AV node, and they are held there momentarily. And that allows the atria to fully empty of blood before the ventricles contract. So, once they leave the atrium, they are held, as I said, they're held there for a little while, and they leave the AV node, and they go down a tiny little space in that non-conductive tissue, and it really is the only route for them to go through. They can't go anywhere else. They'll go from the AV node down the bundle of His. So we have started at the SA node. We've travelled all across the atria. We've been collected at the AV node momentarily, so there's atria to empty. And now that electrical impulse is going to pass through that bundle of His. It's going to travel all the way down the septum of the heart, right to the bottom to the apex. It's going to travel back up the ventricles again, up the Purkinje tissue, basically, the Purkinje fibres. And this basically causes those ventricles to contract from the bottom up. So you're squeezing the heart like a tube of toothpaste. So you're squeezing the heart, and the blood is coming up and out of those ventricles through those semilunar valves and out for the arteries, so the lungs and around the body. Okay, so to go through that again, SA node impulse initiated, travels all over the atria, collected at the AV node, held there momentarily, passes through the bundle of His, down to the bottom of the heart, the Purkinje fibres, and then back up the ventricles, causing the heart to contract and push blood up and out of the heart. Okay? Now, have a go at this table. I want you to kind of link what is happening with the cardiac impulses to kind of what's going on in the cardiac cycle. So as the SA node initiates its impulse that's spread across the atria, what part of the cardiac cycle is that? Pause the video, have a go. So SA node initiates impulses and they spread across the atria. Well, that is going to be atrial systole, isn't it? That's going to be the atria contracting. Those impulses are going to travel across the atria, and it's going to cause them to contract and squeeze and push blood down into the ventricles. Slight delay at the AV node. All that is to allow time for the atria to empty completely. Impulses pass down the bundle of His to the apex. So that means your ventricles are going to contract from bottom up to squeeze the blood out. And impulses up the Purkinje fibres, so that's going to be ventricular systole. So as they pass up, you're going to get your ventricles contracting and squeezing blood from the bottom of the heart up and out. Like I can't even think, like you squeezing a Coke bottle or something and it all shooting out the top. Okay, exam question. So, the heart controls and coordinates the regular contractions of atria and ventricles. Describe how. So it's what I've just talked to you about to basically string it together into a description. It's literally a say what you see question. Describe how does it do it? What are the steps? Have a go. Okay, so answer. 1 mark per bullet point. So we've got a SA node passing the impulse to the AV node to the bundle of His to the Purkinje fibres. Okay, that's the pattern. You're going to get marks if you talked about impulses, electrical activity, cardiac impulses flowing over the atria. 1 mark. Atria contract. 2nd marking point. Mention that band of non-conductive tissue between the atria and the ventricles. That will get you a mark. Talking about there being a delay at the AV node to make sure those atria empty, or conversely ventricles fill. And then we've got our ventricles contracting from bottom up. So ventricles contracting from apex upward would get you a mark there. Okay, remember, if you've got your revision guide, use it. That's what it's there for. That's there to help you. Okay, moving on. So how is all this controlled? Well, it's your brain, obviously, because your brain controls most things. And an area of your brain called the medulla oblongata. And we can see it right here. There it is, medulla oblongata. It's right there, right at the bottom of your brainstem. You can see it right there. And you can see the areas called the cardiovascular centres there, and respiratory centres there that we'll come back to. Okay, and also we'll be talking about the hypothalamus as we go along as well. But basically, within the medulla oblongata, there are two centres that are linked directly to sinoatrial node by nerves. So a nerve runs all the way from here, all the way to your heart. One of them increases it, one of them decreases it. So the increasing, or the one that increases your heart rate, is called the accelerator nerve, which I think is handily named because it accelerates your heart rate. And that's part of your sympathetic nervous system. And then the one that decreases your heart rate is called the vagus nerve, or the parasympathetic nervous system. Remember, your sympathetic's all about getting you ready. It's fight or flight, it's for you to run off and that sort of thing. Whereas the parasympathetic is bringing things back to normal. Now, this diagram here is in the handouts. And again, if you happen to have a CGP book, you might want to use that to help you label it. Try doing that first. If you don't have access to that, you can use this video. You might want to just get the PowerPoint off the VLE. It might be easier doing it straight from the PowerPoint in this video, but that's up to you. But I want you to add labels to it, basically, and here are the labels. Brain at the top, hopefully you'll identify that. Heart at the bottom, hopefully it wasn't too much of a problem. And then we've got our two centres, centre that increases our heart rate, centre that decreases our heart rate. This whole section here is the spinal cord. Then we've got a sympathetic nerve that increases and a parasympathetic that decreases. And you can see here that we've got some receptors here, pressure and chemoreceptors. Pressure receptor is the same thing as a baroreceptor. So baro and pressure are the same thing, more or less, for this level, what we need to know. Medulla oblongata, that's there. And then we can see we're also getting some information here from carotid artery. That's the artery that goes up to your sort of neck in your head. From your head to your neck. From your neck to your head, sorry. Now, so carotid arteries are there. And they've got receptors as well that feed information to the brain. And also you've got receptors that feed information from the aortic arch. So it kind of lets the brain know what's the pressure like of blood leaving the heart, directly from the heart. And then what we've done here is we've just labelled up the heart here and here. You can see that's nice and straightforward. These are the important ones. Your sympathetic nerve that increases your heart rate, and your parasympathetic nerve that decreases the heart rate. Both joining directly onto that sinoatrial node. So they can increase or decrease the number of cardiac impulses that sinoatrial node releases. If you want to increase your heart rate, you release more impulses from the SA node. Decrease the heart rate, release less. Now, we've talked about the receptors here and some more receptors here, so let's look at those. We have two types, our chemo or our baro or pressure. Chemo look at chemical changes. Basically, they look at the levels of CO2 in your blood, dissolved as carbonic acid. And they are in the carotid artery. They're in other places as well. They're not just in the carotid artery. They're in the aortic arch as well. But the purpose of this that we need to know, they're in the carotid artery, and they are inarticulately told you a minute ago, it's the artery in your neck feeding up to your brain there. And as it says there, they're sensitive to changes in pH. So carbon dioxide concentrations, remember, it dissolves into your blood. As I said, it forms an acid, so it's going to decrease the pH slightly, which will be detected by those chemoreceptors. And then we have baro or pressure receptors, and as the name implies, they detect changes in pressure in the carotid artery and in the aorta. Okay, so these two types of receptors feed information up to the brain, and based on what they're telling the brain, depends if you increase or decrease your heart rate. So, for instance, if you are exercising, the amount of CO2 in your blood is going to increase because you're carrying out greater levels of respiration in your muscle cells as they're contracting. So there's going to be more CO2, so your pH is going to fall. This is going to be detected by the chemoreceptors and so on. Okay, if you're exercising, again, because of the changes, you're going to have a higher heart rate, and that pressure is going to be detected. So what I've done is, you may wish to maybe change. I mean, mine's in a table, but you may not like that. When you're taking notes, you might want to change it, maybe a flow chart or something. But basically, what I've done is what happens when you increase heart rate. So perhaps if you're going to start exercising, and then decrease heart rate when you stop exercising. So increase heart rate. those chemoreceptors detect a low pH, and those baroreceptors detect low BP. Okay, impulse is sent to the medulla. There is an impulse sent down the sympathetic neuron to the accelerator nerve, basically to speed up heart rate, but there's none down the parasympathetic. Now, the neurotransmitter noradrenaline is released at the end of the sympathetic neuron. Noradrenaline is released and it binds onto receptors on the sinoatrial node. There'll be no acetylcholine and your heart rate will speed up. Okay, and the other way around. So when you decrease your heart rate, there's high levels of pH detected by the chemos, high blood pressure detected by the baroreceptors. Again, impulses will be sent to the medulla. This time, sympathetic neuron is not stimulated. It is the parasympathetic that is stimulated. There's no noradrenaline. This time you release acetylcholine instead, and you slow down your heart rate. Okay, so again, if you've got the CGP book, remember to read through that as well. Do it in combination with this PowerPoint. Now, describe the role of the nervous system in modifying the heart rate in response to exercise. So pause video, have a think about this table, have a think about, well, what's the role of the nervous system in modifying the heart rate in response to exercise? So if somebody starts to exercise, what's going to have to happen? You're going to want to increase your heart rate. How do you increase your heart rate? Okay, so pause the video, have a go at that. Okay, so the first thing that's going to happen is you're going to increase respiration in your muscle cells because you're exercising. As we increase levels of CO2, the pH of the blood is going to decrease. Oh, come out one here. Therefore, this is going to be detected by the chemoreceptors in your aorta or carotid artery. These are going to send impulses to the medulla, and an impulse will come back along the sympathetic pathway. Noradrenaline is going to bind to receptors on the sinoatrial node, and your heart rate is going to speed up. Okay, so it's describe the role. What happens? What are the steps in increasing the heart rate? What role does the nervous system play in increasing the heart rate? So you've talked about your receptors there, you've talked about impulses to the brain, and you've talked about impulses coming back out along the sympathetic pathway, and you've said kind of what happens in the heart there. So to summarise, things to remember, let's put this all at once. Increase in heart rate, sympathetic neuron, noradrenaline. Decrease in your heart rate is the parasympathetic neuron and acetylcholine. Main important things to remember. Okay, moving on. Exercise, heart and exercise key terms. Now, what I would like you to do is there is a link up on the VLE where you are to post, it says post work for today, basically, and this is what I want. What I want from you is what are the definitions of all these things? What does it all mean? Give me a definition, basically. And then when you've done that and you've defined that, I want you to look at the handout that's also on the VLE. It's under the whole handout for the whole PowerPoint. And it's called Effective Exercise on Cardiac Output. I want you to complete that as well. So those are the two things I want you to submit to me. We've got another video. I see you again on Friday, don't I? So that needs to be with me basically by then, but as soon as possible basically. Okay, so that's the work for you to do. Definitions and complete that. Now, moving on, ECG. I'm sure you're all aware of what an ECG is. You've heard of ECGs before. If you're watching kind of Holby or that, you'll be aware of the sticking pads on people. It's measuring that electrical activity. So it's an electrocardiogram. It causes electroactivity during a cardiac cycle. It lets people know what's going on in your heart. And I'm sure we've all seen the trace before. This should be familiar to you here. Again, there is, on the handout, you can annotate it. So we've got the thing called the P wave here, a little bump there, the P wave. Then we've got QRS, and then that is known together as a QRS complex there, that little bit there, and then we end off with the T wave there. Okay, so that is the P wave there. You can see it there on the scan there. QRS complex, T wave, and then we've got the ST segment there. Okay, so you can see those things on the trace. And that little bit from there is called the PR interval. So between those 2 points is the PR interval. Okay, so if you happen to have some resources to hand, you could look these up, pause the video, look them up, fill this in, what all the bits and pieces mean. Well, P wave is depolarization of the atria, so that's kind of atrial systole. PR interval is as the impulse travels from the SAN across the atria to the ventricles via the AVN node, so it's that little bit there. QRS is ventricular systole, so depolarisation, squeezing those ventricles. T-wave is it's diastole basically, repolarisation of the ventricles, diastole. And then lastly, ST segment. It's kind of resetting, so you've got a little bit of time when you can't sort of further stimulate the heart muscle. Okay, then you've got to hand out what does an ECG show, and that should be on Moodle VLE for you to access with answers. So have a go at that, please. Now, 3 questions that I want you to have a go at answering for me, and the answer in a couple of minutes. So pause the video and have a go at these 3, please. Answers. So, question 1, what causes the release of hormone adrenaline? Well, it's stressful events. If you're fear, excited, shock, panic, if you're into, like, I don't know, horror movies, that sort of thing, something scares you. What's the effect of adrenaline on the heart? Well, it stimulates the sinoatrial node and increases your heart rate, basically. So that's that one. And then question 3, what is the effect of adrenaline on arterioles? It causes them to dilate, so therefore you get more blood to skeletal muscles, so therefore you can get more aerobic respiration, so they can contract faster and you can run away. And you can constrict the arterial supplying blood to the digestive system, so you don't worry about digesting your food while you're trying to worry for the big scary monster. Okay, so have a go at the handouts that have been attached on the VLE. Post your work on that link I've put up there, and I will see you Friday.